this session. Thank you. Very good evening and distinguished guest, co-chairman, chairman of this session and all the learned participants, I welcome everyone and first of all I would like to thank Dr. Kothari and Dr. Vyas to invite me as a lecture and sharing IFCO's contribution for developing NEEM and NEEM coated urea as a organic and agriculture. In the morning, we have discussed a lot of issues for the health issues, how we can use the NEEM for the health purpose. Since IFCO is the largest fertilizer producers in India and we are the second world largest cooperative. So we are the leader of fertilizer. So when we started this using urea, government has thought why we have not using this NEEM as a NEEM coated urea to negative use of urea in the industry. So I would like to make a presentation briefly. Everyone, ये कैसे होते हैं? How we remove? So NEEM is widely used as a village pharmacy and everybody knows how we are using NEEM in each way of life. और मैं तो ये कहता हूँ पहले कहते थे कि नीम हकीम खतरे जान, अब मैं कहता हूँ नीम सबसे अच्छी चीज़. So I think nowadays NEEM is a very important way of using in our healthy life. So which NEEM has several contents and these are the contents which are used for different purposes like human health, for crop and other things. NEEM is used for various purposes. These are the industrial use. Azadirachta indica is a major component which is used for insecticide control and other things, indigenous use. So these are the most of the common use used by the farmers as well as industry. These are some biopesticides are also. Nowadays everyone is talking for the organic farming, so people are not able to get the organic products which are without any biopesticides. So biopesticide is now a emerging need of the organic products. So on several organic fertilizers, teeth cleaners, so these are the different uses of NEEM which we are using in daily life. Urea is a, because since I am dealing with the fertilizer company, so urea is the highest fertilizer consumption fertilizer which produce nitrogen. Because every bag of urea, government is bearing around 2,000 rupees per bag as a subsidy. So looking to all these things around 1.5 crore lakhs rupees is going to subsidize for NEEM this urea to the farmers. So this trial was discussed before, I think 35 years back. Then NFL was started on NEEM coating urea, but that time it was not recognized. But when this 2015, it was mandatory to use all the urea which is produced in India as well as the imported from the NEEM coated NEEM oil. So the nowadays importance of NEEM is coming very fast. Uh, Urea is comparatively cheap because farmers are getting the 300 rupees a bag of 45 kg of bag. So this is cheaper than the salt. So they are highly used, whether it's required or not required, but they are using in uh, it's a different. They are basically misusing the chemical fertilizers. When we applied the urea in the soils, it takes 10 to 15 days to hydrolyze and to take the nitrogen to the plants. So this is the process of urea, and this is the urea used mostly more than 60% fertilizer used as a urea in the agriculture sector. Other DAP and other phosphate complex fertilizers are very less. So government has taken different steps how to increase the use efficiency of urea. So I think 10 or 15 years, some people, if they are related to industry, fertilizer industry, so we have urea super granules, 1 gram of urea, because now this urea is available as a prilled urea or granular urea. So that was the 1 gram of urea was available as a super granule. Then granular urea now is a neem oil coated urea is available. So these are the neem oil contents: azadirachtin, nimine, selenin. We are listening to since morning. So these are the major components which are used as human as per the agriculture purpose also. Industrial use, looking to the use of the urea in chemical industries. So government has taken decision, no urea will be produced without coating of neem oil. So now it's mandatory, every industry, every plant are using neem oil for coating of urea. So now this is issue, this much quantity of requirement of neem coating is not available, neem oil is not available. So looking to this thought, because it has several benefits, neem coating urea slow release करता है, and it also improve the soil health also, and जो bio health है soil की, that is also improved. So one of the thing why we will when we use this neem oil, so there are malpractices are also going because neem oil was not sufficiently available. So people are using different crude oil and other things, but there was no parameter to test whether the neem oil use was used in the coating of neem urea or not. So there was lot of things were happening. So IFCO always thought to how to improve the system, and so when we look this matter, we also produce the urea in different locations. So these are the benefits: increase the yield and other things, but I will just quickly go through the other things. IFCO, how we have addressed these issues to make available more quantity of quality neem oil. So we have we have distributed more than 1 crore neem saplings to the farmer across the country. But these seedlings was not certified. We have purchased from the local nurseries. There was not certified, but we promoted the farmers to plant tree at their field as a one farming. So this initiatives we have done, and we also done one more initiative. We have set up one neem extraction oil plant at our Phulpur, UP, Allahabad. So nearby the area, we have collected the neem seed from the farmer, directly from the farmers, or SSG, which was formed under different programs. So we have collected seed directly from the farmers to run this neem oil plant. We thought we have collected quality. We have not collected the quality material. We pressed it and finalized. Then we see hardly 3 to 4%. The oil was very less than 20 or 25% was recovered. And same time, azadirachtin was very poor. So our management thought, why we not should work for the improving of the cultivars. So this project was started with Forest Research Institute, Dehradun. With the help, we should develop the high quality of varieties which will be available for the future. So we have collected more than 400 germ flower across the country, and we have conducted multi-location trials. Uh, so out of 6 years evaluations, we have identified 6 varieties which were very good for the north and central part of the India. So these are the 6 varieties which are having high content of azadirachtin. We were focusing on the oil and azadirachtin only because we are thinking to have a neem oil for urea coating and other things also. So we kept two things as a major part. So oil, then second is this azadirachtin content. Second part, the flowering. Generally, these local tree which you see, they takes 4 to 5 years for the flowering and fruiting. So this variety, we FRI IFCO 1. So this is having 43.5% oil content, azadirachtin more than 9,500 ppm. Age of flowering is 2 years. It start 2 years after planting of tree. It start within the 2 year. Second variety was released, say out of 400 germplasm. So it is having 35.4% oil content, 9,760 ppm azadirachtin content, and same starting flowering in 2 years. This FRI IFCO 3 varieties, again it was 36.57% azadirachtin content, 6,932 ppm azadirachtin content, and flowering started within 2 years. And FRI IFCO 4, 35.52 per oil percentage, 9,755 ppm azadirachtin content, flowering started within 2 years. And again, fifth variety is 39.75% oil content, 6,000 ppm azadirachtin content, and flowering started within 2 years. And the number of variety which FRI IFCO 6, this is 39.85% oil content, 9,000 ppm azadirachtin content, and started 2 years. So when I was asking question to the previous session, so how we are ensuring this same character goes to the next progeny. So we are not using seed as a multiplication, so we are using tissue culture and cloning propagation. So that will be available commercial production, and that will be available to the farmer, because when we collected the seed and it is not confirmed that same character will go to the next progeny. So we are doing this tissue culture and clonal propagation with the help of different outsourcing the agency. So we are ensuring. We are also working on ahead. We are thinking to have a dwarf variety. Now we are started working the dwarfing of this NEEM varieties, because the seed collection is a very important issue. Because if the farmer, if you convince to the farmer, if you give the seedling to the farmer, I think they are least important to plant there as a crop. Just if you give the plant, they don't even take care. So we are trying how we can use this dwarf variety as a crop, as NEEM as a crop. For the shorter period, they can easily collect. So we are working on that, and hopefully we will have a same kind of content in these smaller varieties, and I think that will be a boom to the agriculture sector and industry also. So these are some photographs which we have conducted during trials. This is about the release committee meeting, and this is about all my presentations. Thank you very much. If you have any queries, so you are welcome.
Thank you very much, Dr. Yadav. You have saved our at least 6 minutes of our time, and it's open for discussion now.
Is it open to questions?
Open for discussion now.
I'm Dushyant from Nico Orgo. Very interesting that you've done these 6 varieties. Have you tried canopy management in NEEM like in the mangoes now, there's canopy management where you trim the tree, so we can go in for closer, dense plantation, and then maybe seed collection also becomes easy.
No, no, actually, every project has a limitation. So our focus was having high quality, high oil content and azotaric content and early flowering. These were the three parameters which we have taken. But now we are working for the dwarfing. So dwarfing purpose, I think they are using different techniques, not this pruning and other things. So that is under process. We will share once it is through.
Okay, thank you.
Any other?
Dr. Yadavji, I would like to know, if the whole country wants to use quality neem coated urea, what
Yes.
will be the total requirement of oil, quality oil for the entire country's urea requirement?
Yes.
Any idea?
I think we are short in neem oil. Around 66% neem oil is available, and we are dependent on 37 from the China.
So we are importing neem oil from China.
Yes, synthetic neem oil also.
And is there national specification for this neem oil? Which
So
has to be put-
there are a lot of queries because there is no specifications.
Yeah, but why
Earlier
not,
it was
as a
started
big
different.
industry, you can develop one specification for neem oil, right?
Right, now it has been standardized to how much neem oil has to be applied for and how much azotaric content also. Earlier it was starting, no, because just they thought we coat the neem oil and it will slow release. Now they are also thinking how we can standardize how much percentage of azotaric in neem oil has to be applied. So now it's working. I think now it's standardized.
So the-
Yeah.
PPM is there.
Yes, now it's standardized. Now it's standardized. Now it's standardized.
Oh, the other question I have is, in your tissue cultured plants, I have no direct publication or evidence. People say that these tissue cultured plants do not flower easily, and fertilization is a big issue.
See, so we have all conducted these trials when we have a two kind of saplings. We have raised seedlings as a clonal propagation and tissue cultures. And we have tested these varieties in the Gujarat. So tissue culture plants are also giving the same flowering and everything is the same as the clonal propagation. And
What-
they are better quality from the seed also.
What is the cost of a tissue cultured plant when it reaches
So
the farmer?
because that was not in commercial forum, now we have started commercial production and hopefully this one sapling of neem will be around 50 rupees. Tissue culture.
Thank you.
It's just a follow-up question. In your paper also you said 22,000 tons of neem oil is required.
Yeah.
And that amount of neem oil is just not available in India at the moment. And then you also mentioned that you've started importing some synthetic oil from China.
Right.
So my question is, if the synthetic oil is coming, then you also are checking it for azadirachtin and other ingredients. So what is the thing which does the nitrification inhibition? Is it the lipid or the oil? Or is it the azadirachtin and the other constituents of it?
Earlier it was only the oil. Now government has set up the standard minimum azotaric content should be required to coat the urea. Earlier it was not finalized. Now it's standardized.
So when you import from China, are you insisting that the synthetic oil has that amount of azadirachtin
No,
in
because
it?
there was the lack of neem oil. So they were using it, but now slowly they are discouraging to import. But that's why we are trying to,
Yeah, so
because
we can-
lot of...
We will
lot-
take it off shelf. Sir, we'll take it offline.
Okay.
Discussion.
Thank you.
Thank you, sir. Thank you, sir.
I am not clear what is synthetic oil.
I think the neem oil, they give the as a synthesize and they add some the local material and use as a synthetic oil.
Oh, I see. Use some other oil as a base and
Right,
then
right.
add other.
Right.
उसके लिए भी तो नीम सीड चाहिए. Extraction कहाँ से होगा?
Right.
No, no,
right.
no. He said about synthetic azadirachtin.
Never successful. Never successful. I have been listening.
Yeah.
Synthesis of azadirachtin, it's a very big molecule. I have known right from the time of Rembold and these international proceedings. And the best thing is to extract from the seeds. They have not been successful.
So that's why we are focusing high quality of planting material is required to cater this need.
That is understood. That is
So
understood.
these
Thank you,
these
sir.
varieties
Thank
will
you.
be commercially available within, I think, next year.
Thank you.
Farmers will get these quality seedlings. Thank you
Doctor,
so much.
thank you very much, doctor. Thank you very much. Very kind of you to give a overview about all the activities.
Thank you.
There's
Thank
a
you so
small
much.
announcement to be made by. Stay back for 1 minute. Yeah, we have to give honorarium for him. No? Yeah. I request Dr. Annadurai to come on stage to felicitate our Dr. RPS Yadav.
Thank you, Dr. Yadav. I am so sorry, Dr. Balaji, I will have to interrupt the technical session for just a brief moment. There is an important announcement that I want to make. We have 2 more lectures. After that lecture, I would like to request all the WNO members to please stay back to attend the WNO meeting. At 7:30, our dinner will be ready. So everyone else, all our guests can proceed towards the dinner after these 2 lectures. But just the WNO members, I request all of them to stay back for the WNO meeting. Thank you, Dr. Balaji. The stage is yours again.
Yeah, thank you very much, Doctor. Now the next speaker will be Dr. Vaibhav Kumar Mehta.
Very good evening, one and all present over here, respected dignitaries and fellow from the academics as well as the industry. First of all, I would like to thank Dr. Nirmala ma'am for providing this opportunity to interact with such a nice crowd who is dealing with one of the important aspect. As we know that NEEM has been like for so many years is a very good product in terms of bioactive ingredients provided by the NEEM tree. So basically whatever technical sessions we are witness since morning, so everybody was discussing about the active ingredient of the NEEM product and their applications in the wide variety of fields. So basically I am not directly associated with the NEEM, but I am indirectly associated with the NEEM product as I have utilized the NEEM leaf extract to prepare the nanomaterials. As we know that India is an agricultural land and we are like facing lots of post-harvest losses. And associated with the agricultural university, one very fine day, our honorable vice chancellor has invited me and he has asked me that whether we can have such kind of solutions with the help of nanomaterials to improve the shelf life of the mango, as mango is one of the important crop in Gujarat. So initially we have started work on the development of biological synthesis of the nanomaterials, as the chemically modified nanomaterials have the implications on the environment and the issue with the biodegradability. So we were looking for the various kind of agro waste and the various kind of like natural products which can be utilized as a reducing agent as well as the functionalizing agent at the same time to produce the biodegradable materials which can be utilized into the post-harvest management of the perishable vegetables and the perishable fruits. So basically these are the domains where the Navsari Agriculture University is working. We are funded with the Department of Agriculture and Farmers Co-Welfare, Government of Gujarat, where we are working with the various domains. First of all, we are utilizing the agricultural waste for the biological synthesis of the nanomaterials. We are also utilizing the nanomaterials as an antimicrobial and antifungal activities. One of the important aspects of our universities is to develop the colorimetric sensors and fluorometric sensors for the pesticide detection at the nanomolar as well as the picomolar and the aptomolar level. We are also developing the nano fertilizers. As we know, IFCO has already come up with the various kind of nano formulations such as the nano urea and the nano urea plus, which are having the 60% of the urea as an active ingredient. So we are also working that how this less amount of urea can be utilized and can be merged and modified with the nanomaterials to give the more yield of the crop. We are also dealing with the use of nanoparticles into the tissue cultural application. We have already provided the scientific information to the agricultural community. We have utilized the silver nanoparticles to reduce the contamination into the grand nain variety of the banana. We are also developing the nano atrazine, which is a nano pesticide for the effective weed management into the maize. So this is the article which was published into the Times of India. So as you can see, the total amount of 92,650 crore value of losses we are facing due to just post-harvest losses. We do not have the proper management of that one. And among that one, fruits and vegetables are facing 4% to the 16% loss. And how the mango? So if we are talking about the mango, then 40 to 50% losses are due to the post-harvest, improper post-harvest management. So by talking into this one of my students has started the work with the development of zinc oxide nanoparticles prepared from the Magnophila indica, that is the mango leaf extract. In that case, we were able to increase the shelf life of the mango up to the 12 days. But still we want to improve the particular kind of shelf life into the days, and we were going to the literature survey and we have come up that we can use these neem leaf extract to synthesize our copper oxide nanoparticles, as the neems are having the very good antibacterial, antifungal and insecticidal properties. So what we have did, we have carried out, we have used the neem leaf extract, we have heated and we have prepared the extract from that one. We have merged that extract with the copper sulfate solutions and heated it up to the 70 degrees for the 3 hours, and we have come up with the copper oxide nanoparticle after the calcination. We have characterized copper nanoparticles with the various kind of characterization techniques that will be discussed in upcoming slides. And one of the important aspect in the mango which causes the disease is the coletotrichum. So we have identified that how the various concentration of copper oxide nanoparticles can be utilized to reduce this particular kind of coletotrichum infection. So this is a methodology by which we have synthesized the copper oxide nanoparticles. The neem leaves were boiled at 60 degrees centigrade for the 1 hour, and the prepared neem leaf extracts were added drop-wise into the 0.1 millimolar of copper sulfate solutions and stirred for the 70 degree for 3 hours. And after that one, repeated centrifugation, we have received the powder, and the received powder was calcined for 400 degree 2 hours to get our final product, that is the copper oxide nanoparticles. The photograph shows that the reactions of the neem leaf extract with the copper sulfate solutions at the various kind of phytochemicals are present, various terpenoids and flavonoids are present, which act as a reducing agent as well as the functionalizing agent at the same time. So here we can see that vial number 4, 5 and 6, which give you the different kind of color with the reaction of NEEM extract and the copper sulfate solutions. So basically when we are talking about the successful completion of the nanomaterial synthesis, we can estimate it primarily with the help of the change of color. So at 180 degree, after 180 minutes, we have successfully synthesized the copper oxide nanoparticles by looking at the color of the solution. So these are the various phytochemicals are present which helped as a reducing agent to prepare the copper oxide nanomaterials. Some flavonoids are present, alkaloids are present, glycosides are present, polyphenols are present, terpenoids are present. We have already given our sample for the GCMS to identify which particular molecule, bioactive molecule is responsible for the reduction of the copper. Further, the synthesized copper oxide nanomaterials were characterized with the electron microscopic image. So here the scanning electron microscope was carried out to just to identify the size and the morphology of the copper oxide nanomaterials, and we have found that the size of the nanomaterials was around like 50 to 60 nanometers. And HRTM was also carried out, and the EDEX analysis just to identify the what was the amount of element copper into the respective copper oxide nanoparticles, and we have identified the weight of the copper was around 51.68%. And the zeta potential was carried out just to identify the stability. The problem with the nanomaterials is to stabilize the nanomaterial for the longer time. So zeta potential is the technique by which we can estimate that how much amount of time your nanoparticle will get stabilized. So here we got the zeta potential value of around minus 32.50 MV, which shows the very good stability of our synthesized nanomaterials. Then we have carried out the FTR analysis just to identify that what kind of metabolites have been utilized for the reduction. So here we can easily see this is the FTR spectrum of the azadirachta indica, and this is the spectrum of the copper oxide nanomaterial synthesized from the neem leaf extracts. So here all the peaks have been reduced, so which confirms that all these metabolites have successfully used as a reducing agent to synthesize the nanomaterials. And the XRD was, I am extremely sorry that that XRD graph is not visible over here. We further carried out the XRD analysis to identify the crystallinity of the material, and it was confirmed that our copper oxide nanoparticle was crystalline in nature. Further, the XPS analysis was carried out, that is the X-ray photoelectron spectroscopy, just to identify whether the copper sulfate has been successfully reduced or not. So based on this XPS spectrum, we confirmed that copper Cu2 plus has been successfully converted into the Cu0, which confirms the synthesis of the nanomaterials. Then here we have used these various concentration of the copper oxide nanoparticles starting from the 50 ppm to the 1000 ppm and with the control as well. So here we can see as we increase the concentration of the copper oxide nanoparticle, we can have a very good zone of inhibition against the collated trochem. So earlier, just with these particular kind of information, we come to know that these particular copper oxide nanoparticles are having a very good antimicrobial and antifungal activity. So this further study was taken for the shelf life improvement of the mango. So what we did, we have taken the various concentration, same as I have mentioned in the earlier slide, starting from the 25 nanometer to the 250 nanometer, and we have deep coated the same amount of mango which was harvested at the same time with the same kind of physiological parameter, and we have studied that particular mango for up to the 16 days. So from the photograph also itself, we can see that at the 0 day all the mangoes are intact. These are the concentration what we have utilized starting from the 25 ppm, 50 ppm, 100 ppm and the 150 ppm. So here we can see the variation into the physiological appearance of the mangoes. The main thing here we can see, this is the control, the last line is the control without any treatment, and here is the mangoes which was treated with the 250 ppm, and here the visual observations can clearly define that how the 250 ppm of copper oxide nanoparticle can be used for the further improvement of the shelf life. These are the % disease incidence with the various concentration and the various treatments. So here at the 250 ppm, we can see that 100% of the disease reduction has been achieved by utilizing, and the shelf life was improved up to 18 days. So it was a remarkable achievement by our university. Further, what is the effect of this particular different treatment on the anthracnose disease? So here the treatment 5, that was the 200 ppm and 250 ppm, which was provided the 100% of the disease management effectivity. And this is a shelf life what we have seen into the photos, that up to 18 days we have successfully improved the shelf life of the mango. So what is the reason that why these particular copper oxide nanoparticles can be utilized? So basically when we are talking about the mango, the ethylene respiration is the most important parameter which will give you the ripening of the respective product. So basically when we are applying the copper oxide nanoparticle, it will like act as a barrier to just go out the gases. So that is why it stops the ethylene respiration, or it can slow down the enzymatic activity as well as the ethylene respiration, which is responsible for the delaying of the ripening, and ultimately we can have the improvement into the shelf life. And just after the coating also, we need to identify, we need to study that what are the various physiological and what are the various biochemical parameters are there. Do we have any kind of impact of copper oxide nanoparticles onto the physiological and the biochemical aspects or not? So that is why we have carried out the various kind of studies in terms of physiological aspect. So first graph is showing the physiological weight loss with the various treatments. So here the T7 treatment is the control one, T5 treatment is 200 ppm, and T6 treatment is the 250 ppm. So here we can see in the control, the weight loss percentage was 25.70%, and in 250 ppm we got 16.29. So weight loss percentage is very much decreased. And when it comes to the firmness, so we need to also check the firmness of the fruit. So here the firmness of the fruit is drastically improved up to 3.08% kilogram per centimeter square, as compared to the control which was only 0.42%. The peel thickness was also estimated, and here we can see there is not that much of difference into the peel thickness of the mango, that was 1.55 and 1.31. So the reason behind that one, so when we applied the copper oxide nanoparticle, it helps into the closing of lenticles on the treated fruits. It generates the barrier against the gas exchanges, specifically the ethylene respiration. The coating efficiently reduces the fruit's metabolic and the enzymatic activities, and it's minimizing the water loss and prevents the softening of the texture and delaying the ripening process. The further studies, the biochemical analysis were also carried out in terms of the total sugar. So whether any sweetness has been decreased in terms of mango or not, and here from the graph we can clearly see that at the treatment of 250 ppm, the total soluble sugar was around 15.22%, as well in the control it was 16.02 due to the more ripening. So there is not that much of difference into the total soluble sugar. What about the titrable acidity? So that one also we have carried out. The titrable acidity was drastically increased, that is 0.320% as compared to the control, that was around 0.107%. And the vitamin C content into the control was 16.20, whereas into the 250 ppm treated mangoes, the vitamin C was 23.57%. So what is the reason behind that all these changes into the biochemical parameters? So because of the treatment of the copper oxide nanoparticles, it slowed down the conversion of the starch into the sugars. It also helps to delay ripening, and higher levels of titrable acidity create an unfavorable environment for many post-harvest pathogens and the slowing down the spoilage. The decrease in fruit acidity, ripening is linked to the reduction of organic acids such as citric acid, which serve as a key substrate for the respiration. And coating further acted as a barrier to oxygen, decreasing the oxidation, that is the oxidative degradation of the ascorbic acid. So this is a graphical representation that how these copper oxide nanoparticles has successfully utilized as a barrier in the gas exchange. So this has provided the barrier for the gas exchange such as oxygen and the water losses. It reduced the respiration and reduced the transpiration, which ultimately gives us the result of the delayed of the ripening. So these are the graphical representation of the uncoated mango and the copper oxide nanoparticles coated mango to conclude my talk. So in the uncoated mango, we can have a post-harvest disease, we can have a fast ripening, we can have a more physiological weight loss. Total soluble sugar and reducing sugar is more as compared to the coated one. Non-reducing sugar is also more. Total soluble solids are also more. When it's come to the shelf life, it is getting decreases. Firmness is getting decreases. Pill thickness is also getting decreases. Titratable acidity and ascorbic acid is much lower as compared to the coated one. So when it comes to the coating with the 250 ppm concentration of copper oxide nanoparticles, the post-harvest is ultimately we have extended up to the 18 days. Ripening is lower down. Physiological weight loss percentage has been lower down. Total sugar and reducing sugar is comparable with the control one. And the shelf life, firmness, pill thickness, and the titratable acidity and ascorbic acidity is significantly improved as compared to the control one. So this is how we can choose the nanotechnology over the chemical management. We can have a targeted delivery in terms of nanotechnology. We need to give the lower dose control to the release system. Environmental impact is less. We have also carried out the copper toxicity study as per the Food Standard Safety Authority of India. When it comes to the mango, the amount of copper should be 30 ppm. But when we have carried out the estimation of copper with the help of atomic absorption spectroscopy, the pulp has shown the concentration of copper was around 10 ppm and the peel was showing the copper concentration was around 9 ppm. So it is under the limit as well. So our work was also highlighted by the news and media, our local newspaper, and the particular work has been like published into the Food Chemistry, which is one of the very good reputed journal. These are our future thrust area at the Navsari Agricultural University. We are further working that how we can utilize the various bioactive ingredient just to control release of pesticide for the effective pest management. We are also developing the microfluidic based devices for the on-site detection of the crop diseases and the shelf life. Further, we are working on the other various products for the shelf life improvement using the various nanomaterials and nanoformulation for the sustainable release of agricultural important nutrients. So I am very much thankful to the Nirmala ma'am, Secretary General of WNO and the organizing committee of WNO Summit for inviting me. Thank you very much.
Yeah, thank you very much, Doctor. We can have one or two questions before we conclude this lecture. Anything is there? Okay. Now I would like to have one discussion. If nobody is taking it up. And Doctor, you mentioned about the particle size as 50 nanometers. What is the percentage of the product in your maybe 1 ml or 1 gram which is having that 50 nanometer particle size?
Basically, sir, when we are estimating the particle size, we are giving the powder. And in that entire image, we are calculating with the image J software is there. So it is considering all the particles available into the image, and that particular software will give us the average diameter.
Correct.
So maybe some of the yes. So maybe some of the particles will be around 90 nanometers as well. Some of the particles
what percentage?
Yes.
What percentage will be under 50?
So maybe
at least under
if
100.
I am talking about 50% are around the 50 nanometers are there.
Thank you, Dr. Weber. Very informative presentation.
Thank you.
Whether any efforts can be made for nanoparticles for stored grain pests?
Actually, sir, recently we have started to see the various use of various biologically synthesized nanomaterials, as our aim is to develop a biodegradable nanomaterials. So when we are synthesizing the biodegradable, it is having the, like what we can say, that it is having a little bit of insecticide activity and pest management. But recently we have started with the same, the azadirachtin functionalized silver nanoparticles, specifically for the pest management.
Thank you.
You compared with control and with your copper oxychloride, I think. Have you compared with other products which are used globally to reduce the post-harvest losses, like one NCP is used, hexanol is used? How does it perform vis-a-vis these products?
Okay. Basically, sir, we have not compared with the product whatever globally are using, but maybe in the future we will do that one as well. So basically now our focus is to incorporate these copper oxide nanoparticles into the certain kind of biodegradable polymer, and our aim is to come up with the bags. So which can be used as a carrier,
yeah.
which can improve the shelf life of
Very
the
good. Very
post-harvest
kind of, very
vegetables as
nice.
well as the fruits.
Very, acha.
Sir, if it when it comes to the nanometers, now lots of universities are working. So I don't have that much of idea that whether any universities are working or not. Thank you.
Thank you, doctor.
Thank you.
I request our friend Nick to come. Okay, Daphne to come on the dais to honor the speaker. Dear friends, the third lecture and the last lecture for this session will be by Dr. Tarun Kant.
A very good evening, respected chair, co-chair, Dr. Vyas, Nirmala ma'am, Dr. Bala, also the delegates from around the country and around the globe, the industry partners, the scientists, and the academia which are sitting here, to all the stakeholders. A very good evening. As we all know, it's for generations, NEEM is present in our landscape. And we all know about NEEM from the childhood times. We see NEEM. NEEM is something everybody knows. Yet, it remains largely unrefined, and particularly when we talk of the industrial supply chain. And we see the tree is everywhere, but the quality part is only seen sporadically. And that is from the industry supply chain point of view I am talking about. So today, as we discuss the blueprint, which I say beyond the blueprint, we are moving away from treating NEEM as a wild resource. Right now, we consider NEEM as a wild resource, not yet probably completely domesticated, a wild resource, and towards managing it as a precious biocrop. So the work that I'm going to share here is a work which has been which transcends several decades, and more recently by a big team of ICFRE organizations. It was a big team effort that we carried out as an all India coordinated project on NEEM, where we did a selection of around 1,000 NEEM trees from 9 different agroclimatic zones. And as we have already been seeing since the morning, the chemical soul of this tree lies in the azadirachtin and the oil. Azadirachtin becomes important from the insecticidal point of view. I'm not talking from the human perspective or from the medicinal point of view. I'm just talking about from the point of view of developing pesticides So the sole from that point of view of this tree is azadirachtin and oil, and there is, as we all know, there is a gap of, this is to meet the massive requirement of 22,000 metric tons. There is a gap of 85% gap in terms of the amount of neem oil that is available. Besides that, I'll be talking, I'll start my talk with a few things. I'll first give you a very brief gist of the legacy work that has been carried out by ICFR and other organizations in the past. Dr. Gurudev Singh sir has already, in fact, touched upon some of the aspects. In fact, we no longer just are looking at the blueprint of NEEM, but we are actually actively engineering NEEM today to become truly a crop which can deliver. So the legacy of genetic improvement of NEEM is basically, it's all started way back in the 1990s. It was the International NEEM Network, which was established in 1994, and it was the FAO and the Danida Forest Seed Center, Denmark, came together, and 23 countries were spotted as the natural or planted regions where the neem is growing. And it was, on a pilot scale seed collection was carried out to begin with. That was the first time a concerted effort was made for neem genetic assessment of its genetic quality and its improvement. It started so way back. And then finally, there was in 1995 a meeting, a survey, an exploration was conducted, and 80 seed sources and natural distributed species were carried out, and 27 seed sources were finally from representing 12 countries were selected, and the provenance trials were established in 15 countries at 20 sites. And in 1996 there was an INN workshop in Bangkok that resulted in seed exchange and nursery productions were addressed. That was a very early days. Guidelines were established in that. And then in 1997 there was a meeting in Myanmar that in which the experts from these groups discussed the establishment and assessment for the future activities of the International NEEM Network. That led to further data recording of the provenance trials, and then finally in India in 2021 we at AFRI organized one data analysis workshop based on the trials, the provenance trials that were established, and to discuss the state of trials. So this was the early work, and that work was that led only to assessment of NEEM in terms of the growth data. Only growth data was discussed. Then this is the state of the trials still existing in India. There were 6 trials laid out at the time, 2 in Jodhpur, 1 in Palanpur, Jaipur, Jabalpur, and then Panampalli in Tamil Nadu. And only 4 trials are still existing with some naturally culled individuals, and this is the big asset we have from the international trials still sitting with us, and some trials were abandoned. That was the International NEEM Network, and then came during the same time AFRI was also working on its own national provenances. 39 seed sources from all over India were collected in 1992, again growth data analysis, but there was no ASA or oil or any biochemical character was studied. Then came the NOVOD board National NEEM Network. That was India's own version of the INN. And in 1999, this was established with different institutions of ICFR and other organizations, all the ICFR institutes plus other institutions. And then as Dr. Gurudev Singh sir has already talked about it, I won't talk much on it. And the ppm range of ASA range from 200 to 16,000 ppm. Very wide variability. Now what happened was, what this program, NOVOD board's NNN project, the recommendations of that project was, it was found that azadirachtin was found to be a highly unstable chemical whose concentration keeps going up and down, and by just doing 1 year analysis and identifying a high ASA plant, we cannot select a variety. So it was argued and it was recommended in that project that at least for 3 years on a larger pan-India scale, a program should be carried out. And that finally led to the establishment of this program. It is the All India Coordinated Research Project on Genetic Improvement of NEEM. In the last WNO summit in 2024 February that was held, I had presented some glimpses, but that was a time when we had not completed the project. Today I'll be presenting basically what we have finally got out of this project. This was funded by the National Authority Campa of Government of India, and in this project you can see the entire team listed on the right hand side. There were 6 ICFRE institutions involved in this, and we have in fact 9 of the agroclimatic zones pan-India that we covered under this program. So it was a massive pan-India program to assess the objectives of this project. This was the core component of the project, where we thought of having, out of these 9 agroclimatic zones, we wanted, and in fact let me also tell you that one of the agroclimatic zone, number 10, was divided into 2, 10A and 10B. So we essentially had 10 agroclimatic zones because 9 plus 1, and we selected 100 neem trees from each agroclimatic zone. That led to total of 1000 trees selected, and we collected 1.5 kg seeds from each during the fruiting period, and there is a very narrow window of 2 months period, as you all know, where we did this. And finally we did oil and azadirachtin under a very stringent set of guidelines that we had. And in fact for developing the SOP for seed collection, because this was a pan-India exercise, so we made sure that every institute, every person involved in this uses the same standardized guideline for collection of seeds, same period collection should be there, and so that we don't, do a work in which there is error. So to reduce the error margin, we did this, and finally that led to collection of seeds and ESA and oil, and through HPLC, of course ESA and oil was done through the succinate method, and the aim was to have the 50 best performers for oil and ESA. There were other project components also, genetic characterization of the best 100 selections through molecular markers. I'll present a little bit on that also. Phenological studies, reproductive biology studies were also there. Then genetic transformation for improvement of gene using the GM technology, introducing foreign gene into the system to improve it, to fast forward the process of genetic improvement. As you all know, the trees have a very long gestation period, and it takes a very, very long time to have a variety developed. So this was a new approach we had, and one of the important aspects was extraction of azadirachtin from cell suspension, that is in vitro studies, which I'll not be talking about today, as there is a dedicated presentation by Dr. Rekha Warrior from IFGTB Coimbatore tomorrow, the first lecture tomorrow morning. So coming to the results now quickly, this is map showing all the neem 1000 neem trees, these dots as you can see, where we selected from different agroclimatic zones, and we tried to reach to these every 1 year. Every 1 year starting from 22, 23, 24, we collected seeds, same methodology, going to the same neem tree and collecting the seed and doing its ESA and oil. So 3 consecutive years of ESA and oil, so that we have a very robust, statistically correct set of data. Though we had challenges also, because when we started doing this work during Corona time and as it progressed, the problem was that as second year we found that some of the trees, we had mortality, or somebody has lobbed the tree, we were not having the seeds. So we had some mortality or some issue. So this is what it looked like sometimes, that a tree is lobbed, we are not having any seeds, fire is there, or some mortality. But nonetheless, we did the selections. You can see these are some of the selections, first year, second year, third year. Selections were done in the first year itself, all 1000, but 3 years data, the pictures from 3 different years I am showing you. These are our CPTs. Then this is a very standard procedure, and which was in fact, which we, the guidance for this we got from Dr. Gurudev Singh sir, and he was our guide for development of this SOP for collection of seed, and we didn't collect anything which was fallen down. We collected the seeds, processed them, depulped them, and as you can see in some of the slides, you can see, still see the variation. In the next one you can see, we dried them in shade, then finally packed them up and sent it to these plastic bags, are the amounts to be sent for the estimation of for HPLC. And this was done by Dr. Nutan Kaushik herself, because the best process is by Dr. Nutan Kaushik. There is a paper, Kaushik et al. Kaushik, in fact, Kaushik 2012. That is her paper. She has developed the HPLC methodology. So in fact, she did our HPLC for all the 3 years. On the same machine, this entire work was done. So there was no way that there could be any error. So we made sure that there should be no error. These are the work done at MIT University. Since Dr. Nutan Kaushik has moved to MIT University, we had this thing, and we used to visit the lab, and this work was carried out 3 years in a row. This is the data, as you can see here. This is, you can see from this, the agroclimatic zone wise, 3, 4, 7, 8, 9, 10A, 10B, 11, 12, 13, you can see. And you see the azadirachtin is highly variable. First year we got very low from the same trees. In the second year we were having much higher aza, the blue lines, and the green lines show very, very high, but very low, very high lows and very high highs. So extremes were very high. And we also found that this is one of the reasons why azadirachtin alone cannot be a way of selecting the high yielding trees. We also did the oil, and before that you can see this small map of India, in which the best, because one of the objectives of this project was not just finding the best trees, but also to find out which is the best agroclimatic zone, which are the 2 best agroclimatic zone. And we found that the zone 10, that is southern plateau and hills, and the zone agroclimatic zone 11, that is east coast plains and hills, were giving the highest aza out of the entire selection. And this is some of the datas, individual datas of that you will see a lot of variation is there. Some of the high yielders in the green. Then oil we again did for this to find the best superior genotypes, and not just oil. In terms of azadirachtin, we did select the 100 best superior, and you can see here the white spots are for oil, and the black spots here you can see are the ones which how they are scattered in the country. High azadirachtin are the black dots on the map. And then we also did one more thing, because we were working with 9 agroclimatic zones. Agroclimatic zone also we've tried to figure out the 5 best performing trees in every agroclimatic zone. So this is that data. And for the gentleman from Gujarat who was asking what about Gujarat, to you sir, you can see the last batch here. We had at least 2 trees which are having more than 5,000 ppm. So 5,000 is the gold standard. That is a benchmark. Beyond anything above 5,000 ppm is considered as high aza. So we had 2 in Gujarat also. So that is an indication that at least 40%. Then we did oil estimations, oil extractions. You can see the oil extractions were done. And here you can see again, this is the process of oil extraction, and this slide also captures the variation in the different seed lots from different regions you can see. And then this data. Now compare this data to the data of aza. You'll find that there is much lesser variation here in this, because you see the graph is much more stable, much more centralized. The trend line is very much straighter. So oil is a much more stable character for selection of a high yielding tree than aza is. Aza is too much of fluctuation. And we also found the fluctuations, we tried to look at the meteorological data also. And when we looked at the meteorological data in 2022, we had a very severe heat wave. Starting from first, I still remember 1st of March, I was walking in the morning, and we were feeling that there is effect of heat wave, the Lu was there. And within 15 days I saw that was the peak flowering time, and the flowers were withering away, and there was a very poor seed set. It was very difficult for our team to collect 1.5 kilogram seeds from even one tree. So this kind of thing happened, and there is a correlation between high moisture levels, the edaphic factors, as well as the temperatures and the genetic factors. So environmental factor is playing a major role in aza, how aza is. And that's why the agroclimatic zone of the South India, where moisture is high, though temperatures may also be high, is more suitable. So if we have to do selections and do collections for further improvement, we should do it in that Southern Indian contingent for aza. But if you look at this map, you will see that the stars, the 3 stars are now up to the central India. So the agroclimatic zone number 10B is the best. 8 and 9 are also equally good for oil. And this is our data on oil. Now this graph in fact shows just the distribution of high oil yielders. and it is pretty much spread out. And this is again for the entire 5 best performers in every agroclimatic zone. Now quickly I'll come to, this was the basic work, the most important work that led to the selection of the CPTs around India, but we also did the genetic characterization of the high yielders. We used a molecular marker system which is known as the Scott marker system. Scott markers is the start codon targeted markers, because we were trying to see something where is in the expression region of where the expression takes place, because we are working with something that is very much a characteristic we are looking for. So we started with some 63 markers. Finally, we had 15 markers we finalized. This is the methodology of DNA marker study that we did on our top performers only. And these were the marker sequences that Scott markers we used. And finally we used, and this is the UPGMA dendrogram, and this UPGMA dendrogram basically tells us that it separates out the populations into 2 large clusters. If you see, there are only 2 major clusters, though there are many subclusters, but we found that the accessions from the same agroclimatic zones often grouped together. Same agroclimatic zones were forming a same subgroup, and suggested that the local adaptations and the environmental conditions influence the chemical expression. That was one important finding from this. We also found that the zone 13 and zone 10A formed a very highly cohesive group. So, I mean, there is some similarity, genetic, they were closely similar, and the 10A and 10B, they were forming the highly cohesive group. And then there were also contrast, the zone 3, 7, 3 and 4 showed varying degrees of overlap, reflecting shared chemical profiles. But the most important was this PCOA analysis, that is the principal coordinate analysis. And if you can see here, it clearly shows that the analysis, the plot clearly reveals that the 9 agroclimatic zones have a structure. There is a central zone, you can see the bigger one, in which you will find that the large central cluster, it contains the accessions from multiple zones. Representing that the genotypes with balanced oil in azadirachtin, and if you do a multi-locational trial, my hunch is that they will perform better anywhere in the India. But if you look at that, the 10A on top, the 10A genotype from this zone formed a compact group characterized by high azadirachtin. This was the high azadirachtin group, and there is a consistent uniformity in it. It forms a separate group totally there. So this was a heartening thing to see, because it is very much in tune with what we found in our AZA and oil data. Quickly, I will wind up in next 1 or 2 minutes, just 1 or 2 minutes. We also worked on the fast tracking of genetic improvement through direct gene transfer, because gene transfer has got very high possibilities of fast tracking the improvement. And this is, I'll take you quickly through the steps of gene transfer. This is the tissue culture protocol, and tissue culture protocol gives out, tissue culture protocol is well worked out, there is no problem. There's no problem of trees, there is nothing like trees won't flower if it is done from tissue culture. There is no morphological problem with the tissue culture. So this is our tissue culture protocol, which we fine-tuned for our gene transfer protocol. This is the gene transfer. This animation shows how the agrobacterium we used, how it uses a small segment of the tDNA goes into the nucleus of the plant, and here the Petri plates show co-cultivation. The NEEM callus is there, and the culture, you can see the film of agrobacterium growing around it. Co-cultivation was done. And here this beautiful picture shows the blue coloration that shows the gene is expressing, the GUS gene. This is a histochemical visual expression assay. And as you can see here, during the tissue culture, the plants are forming. In the G and H, you see the small plantlets are forming from callus, entirely uniformly blue. So the gene transfer protocol we have developed at AFRI is working well. The gene is able to get into the system. We confirmed it through molecular analysis through PCR. We did a cold treatment, because our aim was to produce cold hardy NEEM plants through gene transfer using glycoxylase 3 gene. And this also we got some good results. This is a complete protocol in one slide, how all the steps are involved in gene transfer of NEEM. Finally, futuristically, this is our future plan. This is the breeding plan for NEEM, utilizing what we have, the assets of international NEEM provenance trial, the 100 CPTs selected under AICRP, which I have just presented, and the natural populations, forming this long-term plan, and also the molecular breeding plan involving our gene transfer as well as CRISPR-Cas9 genome editing, which we plan to do now. Thank you very much. Thanks a lot.
Now, thank you very much, Doctor.
Thank you.
Now the session is open for 1 or 2 questions.
No, so after NNN, this was the AICRP which I just presented. After NNN was this entire project that I just presented, all this data, this just got completed in September this year, 2025 September. This project just got ended, and we are now moving into the second phase of this project. We are making a second phase of project where, taking leads from this, we'll be doing multi-locational trials. Now we have got the Pan India asset with us, identified assets. We will be taking up all this battery of 100 best genotypes from the country. We'll be putting them in all the other agroclimatic zone and see whether a South Indian high yielder also performs in deserts of Rajasthan or maybe towards colder region. So this is what we are right now planning to do. This is the drawing board. We are on the drawing board. Hopefully within a year's time this should get functional, because this project is ended, and naturally the next project will start in continuity. Yeah, in fact, we are in the process of publishing it. So once it will be published, it is not, I mean, the report has been submitted. The bound volume of the report has been submitted. The final project completion report with all this data and analysis has been already submitted to the ministry, through our council. It has already been sent to Campa and to the ministry. But yes, we are publishing. We are in the process of publishing. Once the published data is available in the public domain, it will be readily accessible.
Sir, I have a question.
Sure. Yeah.
Clarification.
Yeah.
When you say a lot of edaphic factors, change the expression of azadirachtin and oil content, and in that scenario, do you call this as a ecotype or a genotype?
Well, that's quite an interesting question. But before I answer that, let me just tell one thing that we also did. The complete edaphic profiling of all our CPTs. We have also collected, we have done so we have the edaphic data, we have the meteorological data, we have got the genetic data, molecular marker data. So a lot of set of data are there. Now the question whether it's an ecotype or a genotype, I would rather still stick to a genotype, because until we do the genetics and the environmental effect completely nullified, whether multilocational trial will answer this question finally, but I'll call it a genotype.
The second question is, when all of us know that expression of these limonoids is not a single gene character, it's a polygenic character.
True.
And which gene you will transfer, and then your transformed plantlets what they will express.
Okay.
In terms of secondary metabolite pathways.
Right. So let me because of the paucity of time, I didn't go into much depth of the genetic transformation work. That work was carried out at AFRI Jodhpur only. So let me just tell you very quickly about that. We didn't target genetic improvement of any of the genes responsible for oil or azadirachtin pathway, the tri-nor-tetraterpenoid pathway. We didn't touch that at all. Our aim was totally different. We started working on this problem of developing genetic transformation of NEEM protocol because this was a problem reflected and flagged off by the Forest Department of Rajasthan. We have from the NOVOD board, there was a field trial established at this Govindpura in Jaipur, and that area of Govindpura, every year there is a temperature dip of around 2 degrees Celsius for a couple of days, and there is a frost bite, and everybody knows that neem is not a frost hardy plant. Wherever there is cold temperature, it will suffer. There will be dieback, and we found that for many years the high oil in azadirachtin progeny trial we were having in Jaipur, because of cold bite, there was a reduction in there was a dieback, and this for 15 years there was no onset of the reproductive phase. There was no flowering taking place. So the Forest Department said that we are now having a resource that you gave us, but it is not happening. Anything is happening. Can you do anything about the cold hardiness of it? So we developed this protocol in which we transferred 2 genes. Number 1 was glyoxylase 3, and the second one was CDH, choline dehydrogenase, for making the plant cold hardy. And as I had shown in this slide, we did the okay, it's not visible now. Yeah, the cold treatment. We subjected our callus after gene transfer to 0 degrees Celsius for 24 hours, and then we put it back on the regeneration medium, and we found that there was regeneration happening, but in control everything was dead. So this really is a and whether this will mess up the aza in oil, we cannot say, because anyways when we are doing crossing, there is a lot of transfer of background, transfer of DNA is taking place anyways. So I don't think pinpointed gene transfer will have any ill effect on ASA or oil.
Thank you, doctor. Very
Thank you.
kind of you. It is a very good lecture, very good presentation. Now, sorry, I think one more question is being raised by.
Sir, I have a question.
Sir, yeah, Professor Annadurai has said.
I cannot hear you.
Yeah, I have thought.
Okay.
Professor Annadurai has said, you told that it is a genotype for the NEEM. If it is a genotype, is it possible to change from the perennial to annual?
Pardon?
If it is the annual crop, if it is the flowering and fruiting, it's happening for 5 years, 10 years crop, is it possible to bring it to within a year? No. If it is the genotype?
See, everything is a genotype. It doesn't matter the cultural or silvicultural operations by doing architecture of you can do pruning and you can do many more things in it. That doesn't change anything. That doesn't change anything. It doesn't change. Genotype remains a genotype. Genotype is the genetic makeup of it. The total genetic makeup remains the same, but we can play with a lot of factors. We can put it in an environmental condition where it may have much more growth by providing exogenous material to it, or we may put it in a place where it is deprived and it may remain stunted. So
Doctor,
it doesn't matter.
I think you can have a
Sure.
discussion of
Sure,
Thank you.
Thanks a lot.
Yeah.
Sir,
Any
one
other?
small question.
Yeah.
Sir, I want to know what is the lowest side of the azadirachtin in the neem fruit and the highest side?
See, this question, I would say that we cannot say, though I have shown you the slides, the data is with us, but that doesn't make any sense because a plant may have 200 ppm or 2000 ppm in 1 year. Next year, the environmental conditions are conducive. It may, the 2000 may become 3000 or 4000. So it keeps varying. Even it keeps varying during the time of, if you are not collecting the seeds at the right time, if you miss out a narrow window and if you collect the seeds which are slightly more mature, you may lose ASA. If the seeds are lying somewhere and not processed, you may lose ASA. ASA is something very ephemeral, it loses its, so it's very subjective to tell you what is the ASA. Only the right answer for this would be if we could do transcriptomic work and if we could figure out how much mRNA in the triterpenoid pathway, how much mRNA to different genes is being produced. If we can quantify that, then we can have an answer.
Okay.
Thank you.
Sir.
Thank you very much, doctor.
Thanks a lot.
Thank you very much. Now I request Mr. Nick to come to the dais to honor the speaker.
With this, our technical session of today comes to an end. I request everyone present over here to join us on the stage for the group photograph.